Okay. Alright. So protein kinase A is a allosteric enzyme, and it's going to need 4 cyclic AMP molecules as shown in this picture. The allosteric enzyme pKa is comprised of a regulatory unit as well as a catalytic unit. 4 cyclic AMP molecules are going to bind to the regulatory unit right here, which leads to the separation of the regulatory unit from the catalytic unit. So they're going to split right here. The catalytic site is going to become free, and that goes on to catalyze or phosphorylate other things inside this cycle. So remember that we talked about PDE or phosphodiesterase? We said that it can degrade cyclic AMP as well as cyclic GMP. When phosphodiesterase is inhibited by something like Viagra, then CAMP and cyclic GMP are free to cause vasodilation, increase blood flow, and lower blood pressure. So nitrates produce nitric oxide, which increases the cyclic GMP in the body, and this in turn causes vasodilation. So somebody who takes both nitrates and Viagra could possibly have their blood pressure drop well below a safe window. So here we see something called phosphatidylinosyl, and it's going to be seen right here. This is a lipid in the membrane, and it can be broken down by many enzymes such as PLC right here, or phospholipase C, and it can be broken down by PLC into different products like DAG as well as IP3. So DAG and IP3 are both classified as secondary messengers. DAG is lipid soluble, and this is why it's found in the plasma membrane as shown in the picture. Right, for IP3, one of the major roles is to release calcium from the sarcoplasmic reticulum. So PKC, or protein kinase C, right here, is a class of kinases, kind of like pKa, and it can be broken down into 3 different classes: classic, novel, or atypical. The first 2 classes, classic and novel, are affected by DAG. So basically, DAG is going to increase the activity of PKC of these 2 classes, classic and novel. And remember that we said that IP3 right here is going to release calcium? Well, it just so happens that 2 classes of PKC, the classic and the atypical, need calcium in order to become activated. So in other words, DAG and calcium are both products of PLC, or phospholipase C right here, and those 2 products are going to activate PKC, although calcium is more of an indirect product of PLC. So remember when we said that the GPCRs are going to activate G proteins? So there are 4 major categories of G proteins. There's GS, G0, G11, and then G12/13. These are all different G proteins which release different active subunits that do different things. So GS is going to stimulate adenylyl cyclase, that's what the plus sign is for, and then G0 is going to inhibit adenylyl cyclase, which is what the negative sign is for. G11 is going to stimulate PLC, while G12/13 is going to stimulate small G proteins. So a normal G protein is going to be in the plasma membrane, while the small G proteins are going to be located in the cytosol. So here we have the alpha unit of a GPCR, or a G protein, and remember this one is going to be the major activator of the G proteins. while the gamma beta subunits that we never really seem to talk about are considered the minor activators. They have some minor functions like activating or inhibiting adenylyl cyclase, kind of like GS and G0, and then they're also going to activate PLC, kind of like G11, and they can also activate small g proteins just like G12 or 13. So here's a summary of what G protein classes do. So GS, like we said, is going to stimulate the adenylyl cyclase, and then the G0 is going to inhibit adenylyl cyclase. It's going to basically do the exact opposite. So the activation of adenylyl cyclase and cyclic AMP is going to activate pKa, while in G0 it's going to do the exact opposite. It's going to inhibit pKa. For G11 proteins, PLC will be activated, which in turn produces DAG as well as IP3. IP3 is going to go on to release calcium, which then binds to calmodulin. And these two things bound together are going to activate PDE, or the phosphodiesterase. And what PDE does is it inhibits cyclic AMP. So nitric oxide can either be a primary or a secondary messenger. When it is produced by the endothelial cells, it's going to go to the smooth muscles and cause vasorelaxation. And when it does this, it is a primary messenger. So when it's produced by acetylcholine, nitric oxide is going to be a secondary messenger. So now let's talk about another hormone called ANP, or atrial natriuretic peptide. This hormone is going to produce natriuretics, and these are going to help remove sodium from the blood by the kidney. When the body absorbs more sodium, then blood pressure is going to increase over time. So obese people's kidneys do not maintain sodium homeostasis like they normally should. They actually absorb more sodium than normal, which increases the blood pressure. So we commonly see obese people getting prescribed diuretics or natriuretics to flush out the sodium and water, which is going to then lower blood pressure. So this just shows how different enzymes work on the same molecule, but they break down at different places. So all three of these enzymes are broken down by different hormones, but the one we're going to worry about is the PLA2. And so PLA2 is going to break down right here, and it's going to make lysophosphatidylcholine right here, or the 1-acyl, plus a fatty acid. Phosphatidylcholine is going to produce arachidonic acid by the action of PLA2. And here you can see the arachidonic acid. And arachidonic acid is then going to become inflammatory. So inflammation is a good thing because it kills the foreign invaders in the body, like bacteria. When a certain area swells, it's because blood is rushing to that particular area, so red and white blood cells are all coming together to fight off foreign invaders. So anyways, the arachidonic acid is then going to make thromboxane, which is a platelet activation or a blood coagulation agent. It's also going to make prostacyclin, which is a vasodilator, and it's also going to make prostaglandin, which is involved in the inflammation response. So another enzyme to be aware of is the cyclooxygenase right here. And this can be split into two types, COX-1 and COX-2. So aspirin and ibuprofen are both inhibitors of COX-1, and COX-1 is essential for thromboxane formation. And remember, thromboxane is responsible for the platelet and blood coagulation. So both of these drugs, the aspirin and the ibuprofen, are going to treat pain and fever. Basically, they're going to reduce inflammation by blocking arachidonic acid and inhibiting cyclooxygenase. So COX-1 is expressed everywhere in the body, and you really shouldn't take aspirin every single day because it's going to reduce the production of thromboxane, which is going to lead to blood thinning. So when inflammation is not resolved, it becomes chronic, and COX-2 is going to be expressed. COX-2 is called an inducible enzyme because its expression is induced by inflammation, and Celebrex is an inhibitor of COX-2. So basically what I'm trying to get at is that both COX-1 and COX-2 are both enzymes responsible for the formation of prostanoids. The three main groups of prostanoids are the prostaglandin, the prostacyclin, and the thromboxanes, and all three of these are key parts of how inflammation develops. So COX-1 is found all over the body and is generally a good thing because it helps with the formation of platelets in the body, while chronic inflammation is what causes COX-2 to be expressed. Great. Looks like I'll see you guys next time.